Hi folks, welcome to part 2 of our laboratory on writing for biology. And in this part we are going to focus much more on how to write your lab report. So in the previous part we talked about reading a scientific paper, about the vocabulary within that paper, and those are all important points, but ultimately what you're leading towards is writing this lab report. Now, before we move into these tips, I want to emphasize something about what we talked about last time with that primary literature. When you start going to the library, and most of you or all of you will go to the library virtually. As students at UCA, you have access to an excellent website through our library. It's an outstanding staff over there who will help you in any way they can. And what you want to do as you're reading these papers is try to extract information about the topic at hand, learn the vocabulary and incorporate that into your own vocabulary, as we've said before. But here's another important point. You want to pay attention to the writing style, because most of the people who have been in the sciences for a long time and have done a lot of writing learn much about that writing by reading the primary literature. So really pay attention to how these papers are written, and I really want you to get into the library and do some work, find some papers, read those papers, and you will learn a lot by reading those papers, not just about the content, but about the style and how to write scientifically. This is a really important skill for you. All right, so tips for writing a good lab report. Here's something that you've already seen, right? A paper with a really good title. So under the influence of alcohol, the effect of ethanol and methanol on lipid bilayers. So this title tells you something about the experiments that were done. They were testing lipid bilayers, which are foundational to biological membranes, and how these two different alcohols might affect those bilayers. So if you're interested in that, that title draws you in and you'll want to read more. So the title of a paper, the title of a report, should be clear, concise, precise, and engaging. All right? So make it clear, make it concise. It's not going to be 3 sentences long. Make it engaging. Make your title such that the reader wants to read more. So if you did an experiment on the effect of ethanol in beets, well, ethanol in beets as a title is not very informative. What are you really testing? If the beets get drunk? Well, I don't think so. Make sure that your title tells you more. So for example, temperature-induced cell membrane damage in beet cells. Okay, that tells you that you are going to be varying temperature and looking at how that may affect the cell membrane or the plasma membrane in the beet cells. Or high temperatures damage beet cell membranes. Bam! Right? So sometimes the title gives away your story, and that's okay, because then the reader wants to go in and see really what happened. Why? How did high temperatures affect beet cell membranes? Were they exceedingly high? Moderately high? Well, gosh, the only way I'm going to find out is if I read this paper with this really exciting title. All right, so this one is clear, precise, and engaging. All right, so then the next part of your report will be your introduction. And in the introduction, you're going to talk about what the problem or the principle is that you're investigating. So what was the basis for this report? All right, what is it? What does it do? Why is it important? All right, why might temperature effects on membranes be important? All right, how you determine that and how you set the stage for this paper is using background information that you have gleaned from your reading, from the papers that you've read, from your textbook, and you want to write this for the purposes of this report as if you are describing it to someone who might not know about this topic, like your grandmother or grandfather. Okay? So don't start it with the experiment. All right? You want to start off more broadly and then get more specific. All right? So don't start your introduction with your hypothesis and then start talking about what you did. You have to really give some broad background information. Right? Within this introduction, you're going to talk about your experimental organism, why it was a good choice, and perhaps why other organisms might not work as well. All right, so why were beets useful for studying cell membrane damage? Well, I'll tell you, and you already know this from the lab we did on Tuesday, the previous lab, last week, sorry. Beets have colored pigments inside of the cells. If you damage the membranes, those pigments will leak. So you have a way to measure. You can actually use the spectrophotometer to measure that. All right. Why are mice good for genetics experiments? Well, it's a good question. Well, one of the big reasons why mice are good for genetics experiments is that their life cycles are very short. So they can go from conception to birth to maturation to conception again within a very short period of time, so that you can actually do multiple generations within a very reasonable period of time and then study the genetics as the genes are passed through those generations. Okay? In your introduction, you should answer the question of what is your research question and what is your hypothesis. They should be clear in your introduction. So make sure they're clearly stated at the end or near the end of your introduction. All right? Don't just tell what your hypothesis was, provide a rationale for it. All right? That, and all of this should be in your introduction. All right. Here's some examples. All right? So an example introduction. And this is violating one of the rules of slide making, as I've got too much text on here. But give this a read. And you can pause this and read this. But when you read it, you'll see that it's pretty concise and it has all of the components that I was just talking about. So every living cell is surrounded by a plasma membrane, and eukaryotic cells contain other internal membranes. Biological membranes consist of a phospholipid bilayer and contain transport proteins. And then here is a citation. All right, you're citing a source of information that you use to inform this introduction. In this experiment, we examined the effects of incubation temperature on the cell membranes of beets. Beets contain beta-cyanin in their vacuoles, making cell membrane damage easy to detect. Our research question was, how will varying temperature affect cell membranes? Our hypothesis was, if temperature increases, so will membrane damage after a critical temperature is reached. As energy is added to the beet samples, the movement of the phospholipids and proteins in their membranes will increase until the membrane becomes leaky or ruptures. And again, if you need to pause that, read this carefully. This is a really nice, concise introduction and contains all of the components I talked about in the previous slide. All right, then you go into methods. And the methods in your paper will be abbreviated because we cannot do this lab online. We'll be giving you an introduction and some data sets. There'll be a video for you to watch, and then you're going to work with that data. But what you need to think about is, how did the methods that were used differ from what was given in the lab manual? All right, so and there's ways to state that when you write your report. So you want to make sure that you describe things such as the concentration of solutions that you used, the treatments that you applied to your variables, or anything else that you changed that was not specifically outlined in the laboratory manual. All right? Never, never, ever, and I mentioned this once before, but I'll say it again, never describe your methods as a bulleted list. You must write your methods in a narrative form, right, using sentences. So always do that correctly. Here's an example. Procedures were followed as described in the lab manual. Cite the lab manual. This is a proper way. There's 3 authors on the lab manual, so Chowinski et al., 2019, with the following exceptions. Beet cores were cut using a 0.5 centimeter coring tool to provide maximum surface area on the beet segments. Each treatment was completed in triplicate, so n equals 3. Okay? Pretty self-explanatory. All right, results. So this is when you are now going to report the data that you collected in your experiment. So you're going to present the results of each experiment in the form of a correctly formatted and captioned figures. So make sure you use your laboratory manual. This is all described in Appendix C in great detail of how you actually build these figures. And you've read this before, so it should be familiar. You should know it, but go back and read that again. Now, the narrative or written section, all right, will describe trends in the data. And I said this is a required section. So yes, you must have figures and a written narrative. All right, and the data should be described numerically. So you can use numbers such as the slope of a line when you are writing about this so that people know what those numbers mean. All right. If your narrative is a little bit redundant of the figure caption, that's okay. It's not identical, but it can be similar. I'd be surprised if it wasn't. All right. Describe qualitative data. All right. So things that you observed in the data that you didn't put a number to. And make sure when you're writing the narrative that you refer to the figure as I've shown here. Figure 1, maybe figure 1, and later on you talk about figure 2. Make sure you do that. Don't forget. All right. This is not the place where you interpret or talk about if the data was good, bad, whether it supported or refuted your hypothesis. This is not the place. You're simply reporting the results in this section. All right, so an example. This experiment investigated the effects of temperature on beta-cyanin leakage from beet roots. Between 25 and 60 degrees Celsius, there was no significant change in beta-cyanin concentration, figure 1. Leakage increased by 150% between 60 and 65 degrees C and 120% between 65 and 70 degrees C, figure 1. So this is leading you, the reader, to go and look at that figure. Solutions with higher temperatures appeared to be much darker in color than lower concentrations. So this is your qualitative statement. All right. The quantitative statements were derived from collecting data on those different solutions using a spectrophotometer. All right, when you make your figure, excuse me, make sure that you pay attention to all of these different things. So make sure that your axis has even increments. Make sure that you have an axis label and you have proper units, all right, for each one of these axes. You have a dependent variable on the y, independent variable on the x. Your data points should be means or averages, and they should all have error bars, and we've talked about how to do that in Excel. Do not use the automatic standard error calculator in Excel, and don't use the automatic error bar. If I see in any given figure where all the error bars are exactly the same size, that tells me something's wrong, because that's not realistic. And if I see error bars where you've got one which is vertical and then there's also a horizontal error bar, I know that you've just clicked standard error or error bars in Excel, and it puts these crazy bars on there that really don't mean anything. So don't do that. All right. Now, you then have, to make this a figure, you have a caption. So every figure has a number. So this would be figure 1. If you have another one, it'd be figure 2, so on and so forth. You'll have a title. You will have a statement about the data that might give you something, some indicator about the quantitative data which is being presented. And then, and I'm going to move this one, I didn't make this figure, but some people put this mean plus or minus standard error in the middle. I will never do that, so I will move that over here where you'll put mean plus or minus standard error, semicolon, n equals 3 per treatment. So pay attention to that, and I won't dock you for that because there are different ways to do it, but I have a very strong preference for that mean plus or minus standard error to go at the end the way it's written on page 73, which is the Appendix C in your lab manual. All right. Discussion. All right, so this is where you'll have some conclusions, and this is where you will interpret the data. So you will state if the hypothesis was supported by the data, all right, and you can and should refer back to your figures again, so you're going to be using that data. This is where you interpret your results, all right, so this is where you try to explain what happened. All right, the results tell you what happened. This is where you will talk about why you think it may have happened. You will also compare your results to the results which others have previously reported. This is another reason why you will go out and read the literature, not only to build your introduction, but now to put your own results into the proper context. So this is where you can talk about your results are similar to what was reported in other papers by other researchers, or maybe you found a different result. You can only do that if you use your literature effectively, and make sure you cite those papers. Right? And this is also where you can then talk about next steps. So you can talk about what future research you might do, what other experiments could be done to make this project even stronger. All right? All right, so here's an example of a discussion. All right, again, you're always welcome to pause and read. So the hypotheses were supported by the data. Figure 1. As temperature increased past 60 degrees Celsius, cell membrane damage as measured by beta-cyanin leakage also increased. As predicted, it appeared that beets can tolerate a wide range of temperatures. However, as temperatures increase past 60 degrees C, membrane fluidity increases until the membrane integrity is compromised. The leaking of beta-cyanin through the membrane is likely due to the rapid movement of membrane molecules. Give a citation for that, because that's making a pretty strong statement. So Smith and Jones, 2019, have already reported on something like this. Membrane damage occurs at temperatures that are much higher than beets are likely to experience in their natural environment. Okay? So some examples there. Beets are usually planted in early spring or at the end of summer. An experiment that compares the effects of temperature on beetroots along with other plants that grow in hotter temperatures would be useful for agricultural purposes. I predict that some membranes of plants that normally grow in the summer heat will be more resistant to increased temperature than beetroots. All right, so this is giving you an idea of some future experiments and how you think that might turn out. Really, it's giving a rationale. So you're not just pulling this out. So, for example, this is one experimental thing that I used to see 25 years ago or more, where people would say, okay, I'm going to look at beetroots and heat it up in water and see what the effect is on leaking beta-cyanin. And then say, oh, okay, so a future experiment, I want to incubate the beetroots in Coca-Cola. And I'd be like, what? That has no relevance to anything whatsoever. And even if it did show something, are farmers going to soak their fields in Coca-Cola? Yeah, I don't think so. So make sure that when you do this, that you stay relevant, stay real, make sure you're really thinking scientifically. All right, now, references or literature cited. All right. Here is a reference to a textbook that is a secondary source. Here is a reference to the lab manual. Again, a secondary source. Here's a reference to a paper from a journal. Primary source. When you write your report, you must have at least 3 references that you cite in the literature cited section, and one of those must be a primary journal article. Okay? You must have at least one primary article. Don't forget that. You have to go to the library to find those. All right, some additional details about this lab report, and you'll be getting the actual assignment in an upcoming lab soon. When you write this report, you will almost always write in the past tense. Pay attention to that. You've already done the experiment, so it doesn't make sense to write, for instance, an introduction or materials and methods section which is in the future tense. That is, I will do this. No, it has been done. So use past tense. Don't use quotations. All right, you won't see those in your scientific writing. Avoid the use of personal pronouns. I did this. We did this. Now, the Piranha paper uses some, and I've seen this over the past years where the use of personal pronouns in scientific writing is becoming more acceptable. I'm not a fan. It's not typically done, right? And it's just something you want to avoid. Now, when you avoid the use of personal pronouns, it will sometimes cause you to write in a passive voice. You want to try to minimize that, but that's not always bad. That's what I'm trying to say. If you end up in a passive voice and your word processor flags you for using a passive voice, but you're using a style like what you've read in these articles that you're reading, that's okay. All right, write logically and concisely. So make sure you proofread your paper. Avoid saying the same thing more than once. The exception to that is when you're writing, for instance, your results section, your narrative, and the caption for a figure. There may be a little redundancy there. But use your resources again. Get in touch with the Center for Writing and Communication. There are people there that will proofread things for you. Use 0.5-inch margins on all sides of the paper. Times New Roman, font size 12, double spacing, and it will be a 2-page paper including figures. So I suggest that you practice doing things like making your figures smaller and wrapping text around the figures. So check that out. All right, those are all features that you can use to embed a figure within the writing and then wrap the writing around that figure so you can fit it all within 2 pages. All right, what this is saying, proves versus supports. Don't use the word prove when you're talking about your hypothesis and whether your data agreed or supported your hypothesis. That's the word you want to use, that was supported. You're not going to be able to prove anything with these experiments. You're likely not going to disprove anything either, but you can get data that does not support your hypothesis. All right. Ways to improve your scientific writing. And it's about conciseness and eliminating unnecessary words. All right. So you can have things like phrases. It is likely that. And you can cut that down. Just say likely. In the vicinity of. Just say near. It must first be established that. All kinds of wasted words there. Just say first. Due to the fact that is the same as because, so on and so forth. So you get the point. You only have 2 pages. Many times in other disciplines when you're writing, you'll have 10, 20 pages. You have 2 to write this report. And this is real. And this is why you are limited to 2 pages, because you have to be able to convey the information in a very limited number of words. I've often had to take 3 to 5 years of research, 3 to 5 years of research, and submit that for presentation at a meeting in a box that'll only hold 100 words or 75 words. It's not many words to be able to summarize that research, but with practice, it can be done. Okay, so writing styles. All right, here's some examples. So you have an original writing. This is the way somebody might write a lab report. Although I added 5 mLs of 10% ethanol, there was not much color change noticed in the test tube. Okay, it conveys the idea, but it's pretty casual and it's not very succinct. And you can convey more information in fewer words. So you could revise that. Say I added 5% or 5 mLs of 10% ethanol, but did not see a color change. Okay, or adding 5 mLs of 10% ethanol to a beet cylinder did not change the solution color. All right. The third one's the best one. It has fewer words than the first one, but yet it tells you more. It's about conciseness. It's about style. All right. Here's another one. Fertilizer provides very important nutrients to plants for growth. Generally speaking, plants cannot produce their own nutrients. Therefore, they need to obtain them from the environment around them. This experiment is important because just like in the real world, fertilizer will be put on the plant so that we can test whether or not growth will be increased or decreased. All right, so it's not terrible, right? It tells you some information, but let's look at a revised one. Plants cannot produce all the necessary nutrients for growth, therefore they must be obtained from the environment. This experiment simulates fertilizer use in agricultural practices. Our experiment investigates which concentrations of fertilizer are most effective for increased tomato growth. All right, so it's a little bit shorter than the first one, but it actually contains more information, and it's more direct, less casual. All right, here's the picture you've seen before. I'm not going to elaborate on this again, but make sure that even though you are getting feedback from the Writing Center and working on numerous drafts of your reports, make sure that all the reports that you turn in are your own reports. And I will very much appreciate that from each of you. Okay, folks, that's all that we have for today. So I hope this has been useful for you. I hope when we get to writing these lab reports that you are better prepared now than you would have been otherwise. So use your time wisely. I know this has been a short lab exercise, pretty much of a dry lab on how to write lab reports, but this will be important because we have this big report coming up very soon. So with that, I'll end this segment and wish you a good day. Take care, everyone.